Our next speaker will be Dr. Stacia Friedman-Hill. Dr. Friedman-Hill is a program director in the Division of Translational Research at the National Institute of Mental Health. She is the chief of the Mechanisms of Cognitive Dysfunction program and the Trajectories of Neurocognitive Functioning program. So you may have to explain to us what that means here in a moment. Dr. Friedman-Hill has a Ph.D. in neuroscience from the University of California, Davis. After earning her Ph.D., she was a postdoctoral research fellow in the intramural program of NIMH. Upon completion of her fellowship, Dr. Friedman-Hill served as the director of the Cognitive Neuroscience program at the National Science Foundation. In 2009, Dr. Friedman-Hill returned to the NIMH to join the extramural program, and I hope that you can tell us a little bit about some of the particular initiatives that you're working on at this time. Dr. Friedman-Hill.
Thank you for the wonderful introduction, and thank you for the invitation to talk to you today. So I am looking for my slides. There we go. So I was invited to talk about brain development, particularly in elementary school children. And I could talk to you for hours about this, but I only have a few minutes. So what I'm really going to do is just give you a sampling of different methods we have for studying the brain and what some trends in development are over over middle childhood, the age that children are in elementary school. Oh, great. So my first slide was really just an introduction to remind you that in childhood, it's not only the case that children are physically growing an enormous amount, but their minds are developing as well. There's many levels of growth in children's cognitive abilities. And psychologists traditionally have many different conceptual schema for characterizing stages of development of mental processes. But it's only recently that we've been able to really sort of peek inside the head to look at the brain and understand why cognition and emotion and social processing are changing over childhood. And so one of the very first trends that you see when you start to examine neuroimaging data is that the brain does not develop everywhere at the same time. One real finding that has influenced the field is that different regions of the brain peak and develop and mature at different time points. So if you look at the top of this slide, these are schematic diagrams of sort of where the peak maturation of different parts of cortex are. And you can say, for example, motor cortex or sensory areas are maturing much earlier in development in middle childhood than areas like the prefrontal cortex, where they're reaching their maturation levels sometimes as late as the early 20s. And so this is important as well. There are multiple processes that are development. They're not just one process of maturation. Many changes are occurring at the cellular level, at the neural level, and at grosser scales where you can measure the size of regions. Before we delve into some pictures of the brain, we also should consider, because I am coming from the National Institute of Mental Health, what we know about development and mental health disorders. So in this slide, I really wanted to show you that if you just look at the age of onset of different types of mental health disorders, you can see that they vary widely. So as Dr. Murphey mentioned, early in childhood, you see the onset of ADHD or autism, probably reflecting processes that even occur before birth. Whereas as you move into middle childhood and adolescence, you see an increase in anxiety, mood disorders. And then there's some disorders like psychosis or schizophrenia that, although they may be the result of early injury to the brain, really don't have their onset until adulthood. And so just looking at the age of onset gives us some clues as to what some of the neural mechanisms might be that are leading to a risk for mental health disorders. On the right side, I wanted to show you also the difference in prevalence rates. And as was previously mentioned, you can see that there are different risk factors. So boys are more likely to be diagnosed with ADHD or autism, whereas girls are more likely to have an anxiety disorder. And so we should expect that we can see different periods of susceptibility in the brain and differences that are based on sex. What I'm showing you here, and now I'm going to start delving into some pictures of the brain. One way you can look at the brain is to look at anatomy and structure. And so this is a study that was done in the intramural program of NIMH by Jay Geed and his colleagues. Individuals who were age 3 to 30 came in every 2 years and were scanned in an MRI scanner, and that yields anatomical pictures of their brain. And then you can use computers to form these 3D images, and these are colorized. Your brain obviously isn't these colors, but the warm colors are showing areas which have a peak in gray matter volume. The cooler colors are showing decreases in gray matter volume. And this is an example, too, of the heterochronicity of changes. Again, if you look, for example, at some regions that are yellow in the beginning of development, early in development, age 3 or 5, you can start to see how gray matter decreases, and it doesn't decrease at the same rate everywhere. Now I'm going to really start going fast because I'm told I only have a few minutes. So this is other measures that you can pull out from the structure of the brains are looking at white matter, looking at the whole size of the brain. And in this figure, I just wanted to show you that we can find differences between boys and girls on average. So the blue lines, especially the bold blue lines, are the mean of different volumetric measures from boys. The red are different structural measures from girls. And you can see that there are differences across development and differences for different changes. The little arrows on the bottom are showing you where the peak of development is. So in general, boys' brains have higher volume metric measures, but the girls' brains are showing peaks at earlier ages. Okay. Also, when you look at structure, in addition to looking at size of things, the volume of things, you can look at the connectivity. So regions in the brain are connected by the axons of neurons, these fiber tracts. This is an example from the Children's Hospital of Philadelphia. In the top row, those are the brains of male subjects, participants who are age 8 to 22. And that's averaged across all those ages. And the second line is the brains of females. And what this is showing is that there's a change in connectivity patterns. So the male brains are showing more connectivity from different regions that are in the same hemisphere of the brain. The females are showing increased connectivity between the same region but in different hemispheres of the brain. So far I've only been talking about brain structure, but we also have different neuroimaging measures to look at function. So activity in the brain, not just static structure or the hard wiring. We can actually look at changes that reflect the physiology. And so that is called functional MRI. I just wanted to give you a couple of really quick pictures of what you can do with this. The reason it's interesting is that you can actually measure brain activations while the individuals are doing a task. This is an example from Damien Fair. It was a study of children who have ADHD and children who don't. On the left-hand side of the figure, he looked at which parts of the brains were active and correlated with each other while the subjects were just resting, not really doing anything. And the bottom row, line B on the left, shows some differences between the ADHD children and the healthy controls. On the right side, he then asked them to do a task. They did something called delay discounting. Essentially, children are asked, they have to make a choice, and the choice would either be associated with a very small but immediate reward, or it would be associated with a larger reward, but they would have to wait a period of time to get this reward. Children with ADHD typically go for the small immediate rewards. And so a region in the brain which is associated with reward system, the nucleus accumbens, changes its connectivity when with prefrontal cortex, and that's associated with their performance on this kind of reward task. I also just wanted to mention that there are some researchers like Vinod Menon at Stanford who are looking at academic skills and how these are correlated with activations in the brain. He has been looking at reading tasks and mathematical ability. He's found consistent regions in the brain that are specific for reading, other regions which are specific for math, and he finds that children who perform poorer on reading or math, have less activations in these regions, less connectivity between these regions. And the really intriguing finding from Dr. Menon's lab is that he can look at the effects of tutoring. So he had a study where children underwent an 8-week period of one-on-one math tutoring. And after that, he actually found that the regions which were implicated in mathematical problem solving showed an increased connectivity with other regions, and that also increased activation. And then this is the last data slide I have. I also wanted to touch on how the environment can affect brain, both brain structure and brain activations. So this was a study from Elizabeth Sewell at UCLA, and she measured, in a very crude way, socioeconomic status. She was quantifying it by looking at a family's income expressed as a proportion of what the poverty level for a family of that size would be. And she found on the left that certain brain regions like the hippocampus and the amygdala actually were associated with different volumes depending on socioeconomic status. And then on the right, she also shows other regions that there's an interaction of age and development with socioeconomic status. And so in conclusion, what I really wanted to do by showing you this brain imaging data is to suggest that we know a lot about how the brain is changing over time, which regions are changing. We now know which regions of the brain underlie things like attention and memory and learning, and even specific learning like reading or math. And we also have the ability to measure changes over time. And so this might suggest for you regions that might be sensitive for training or interventions, and also special time periods where the brain is changing a lot, and where time periods where we should consider whether there's plasticity, and we should really focus our interventions during particular developmental ages. All right, thank you.